There are photographs of the last one. She is perched on a branch inside a small wire enclosure at the Cincinnati Zoological Garden. Her tail is long and tapered. Her breast is a faded rust orange. Her eyes, which were once described as a deep, living red, look pale in the photograph, though that may be the age of the film. Her name was Martha. She died on the 1st of September, 1914, on a Tuesday afternoon. A zookeeper found her on the floor of her enclosure. She had been the last passenger pigeon on Earth for at least 4 years. Before her, the species had numbered somewhere between 3 and 5 billion individuals. 3 to 5 billion birds. John James Audubon described watching a flock pass over Kentucky for 3 full days. 3 days during which the birds overhead were dense enough to block out the sky like an eclipse. He estimated the column contained more than 1 billion individuals. A nesting colony documented in Wisconsin in 1871 stretched for roughly 150 miles, the distance from New York City to Philadelphia and back, and was estimated to hold more than 136 million birds. The passenger pigeon made up somewhere around 40% of the total bird population of North America. Not 40% of a related genus, 40% of all birds on the continent. 50 years after Audubon's account, there was 1 bird left in a wire cage in Cincinnati. The question that should have had a simple answer, how does a species of 3 to 5 billion individuals disappear inside a single human lifetime, turned out to produce a more complicated scientific argument than expected. Not because the role of hunting was ever in serious doubt. It was the hunting. But when researchers extracted DNA from museum specimens and read the passenger pigeon's genome, what they found did not match a story that had been building in the literature for a decade. That story said the species was already fragile before the first commercial shooter arrived. The genome said it wasn't. Martha's body was packed in a 300-pound block of ice and shipped by train from Cincinnati to Washington, D.C. She arrived at the Smithsonian Institution, where a taxidermist mounted her on a branch in the posture she holds today. She has been at the National Museum of Natural History ever since, more than 100 years, under glass, a few rooms away from specimens collected by naturalists who shot passenger pigeons by the thousands and never imagined there would be a last one. If you're finding this useful, subscribe. This channel makes episodes like this one, and there are more coming. The Smithsonian also held what those 19th century collectors left behind. Museum drawers full of study skins, trays of bones, pinned wings. The specimen record for the passenger pigeon is unusually rich, not because anyone anticipated needing it for genomic research a century later, but because collectors in that era were meticulous about the birds they were shooting in extraordinary numbers. Those drawers are the reason the 2017 study was possible at all. In 2017, a research team led by Gemma Murray at the Australian Centre for Ancient DNA published a paper in the journal Science examining the genomes of 17 passenger pigeon museum specimens drawn from across the species' former range. The paper was titled "Natural Selection Shaped the Rise and Fall of Passenger Pigeon Genomic Diversity." To recover those genomes, the team extracted degraded DNA from dried museum skins and bone fragments, material that in some cases had sat in a drawer for over 120 years. The process requires isolating fragmented genetic material, sequencing each piece, and assembling the results against a reference genome to reconstruct what the living bird's genetic code looked like. It is painstaking work. What it returned overturned a hypothesis that had stood in the literature for years. To understand what Murray et al. Overturned, it helps to know what the earlier work had argued. The prior genomic analysis, published by a team that included Beth Shapiro at UC Santa Cruz, had proposed that the passenger pigeon's population may not always have been the stable, multi-billion bird mass the historical accounts describe. The hypothesis was that the species underwent natural boom and bust population cycles over centuries, expanding and contracting with habitat conditions and food availability. Under this model, the population that European colonists encountered was one peak in a recurring pattern. And a species that had evolved under that kind of instability, the argument went, might carry the genetic marks of it: reduced diversity in certain regions, an accumulation of mildly harmful variants that a population of steady billions would have filtered out. A genome shaped not by sustained abundance, but by the particular pressures of repeated crashes and recoveries. If that were true, the passenger pigeon was not simply a victim of industrialized hunting. It was, in some meaningful biological sense, already pointed toward collapse before the hunters arrived. The Murray study tested this against the actual genomic data. What it found was essentially the opposite. The passenger pigeon's genome showed the signatures of a population that had been large, stable, and evolving under strong, efficient natural selection for a very long time. The pattern of how mutations accumulated and were filtered across generations was consistent with what you would expect from a species that had genuinely been enormous and enduring. Harmful variants were being removed at the rate you would predict for a population of billions. Beneficial variants were spreading the way they do when natural selection has an enormous pool to work with. There were no detectable signs of pre-existing fragility, no bottleneck signatures from the centuries before European contact, no evidence of accumulated genetic damage from repeated boom-bust cycles. The genome looked like what a genome looks like when it belongs to a species that has been large and stable for a very long time, which meant the collapse was not preordained. The species was not biologically en route to extinction before the commercial shooting began. A healthy, stable, numerically vast population was hunted to extinction, systematically, industrially, in roughly 5 decades. The scale of what that required takes a moment to absorb. By the middle of the 19th century, passenger pigeon hunting had become an industrial enterprise. Professional netters using spring-loaded nets could take 50,000 birds in a single day at a productive site. Rail connections allowed nesting colonies to be worked simultaneously by crews of hundreds, with birds shipped live and dead to eastern markets by the barrel. A barrel of passenger pigeons sold for 50 cents at the market peak, a price so low that the volume required to make it profitable was itself a mechanism of extinction. Martha herself weighed approximately 340 grams, a little less than a pound. She was smaller than most domestic pigeons. The last confirmed large nesting colony in Petoskey, Michigan, was worked by hunters in 1878. The last confirmed wild passenger pigeon was shot in Ohio in 1900. Martha died in 1914. The entire collapse, from a species that constituted 40% of North American bird life to zero, took roughly as long as it takes a child born today to finish their education and enter the workforce. The Murray finding matters because it removes an available exit from that history. If the species had been genomically fragile, if the genome had shown a population already in decline, carrying a load of accumulated mutations, already losing the efficiency of natural selection, then the hunting would have been a final push against something already falling. The genome says there was no prior fall. The hunting was the whole story. Before we get to what this finding actually means, and to what else the Murray paper found, subscribe if you want to hear the rest. That distinction carries weight beyond the passenger pigeon. The question of whether a species was already biologically compromised before human activity intensified is asked about dozens of extinctions. It is one of the ways the magnitude of a historical event gets redistributed, partly to climate, partly to habitat change, partly to pre-existing condition. And the passenger pigeon is one of the cases where the genomic record is now clear enough to close that question. There was no pre-existing condition. What happened to this species was a choice made by an industry that could have been made differently. The secondary finding in the Murray study concerns what the passenger pigeon genome demonstrates about the relationship between population size and how efficiently natural selection operates. Population genetics has long predicted that large populations should evolve more efficiently than small ones. More individuals means more opportunities for beneficial mutations to arise and spread, and more rigorous filtering of harmful ones. This is a well-established theoretical prediction. It is, however, difficult to test empirically because most species for which ancient DNA has been recovered were never as numerous as the passenger pigeon. The 17 specimens in the Murray study gave researchers something rare: genomic data across a genuine extreme. The signatures shift in exactly the way the theory predicts, from periods when the population was at its largest to the earlier periods when it was somewhat lower. The passenger pigeon becomes one of the clearest empirical demonstrations of a fundamental evolutionary principle in the entire museum record, not because of what happened to it, but because of what it was. Bob Zink is a professor at the University of Nebraska-Lincoln. He was also the lead author of the earlier genomic study that Murray et al. Were partly contesting. He has said publicly that he does not believe either paper, including the 2017 science study, fully explains the extinction. The rate of decline, he has argued, is still not adequately accounted for by the genetic evidence. How 3 to 5 billion animals disappeared inside 50 years remains, in his assessment, an open scientific question even after 2 rounds of genomic analysis. That is worth noting carefully. Not because it overturns what Murray et al. Found, it doesn't, but because it locates where the science actually sits. One question has been answered: the species was not already fragile. Another question: exactly how the hunting alone, even at industrial scale, erased a species this numerous, this quickly, has not been. The passenger pigeon was a socially colonial bird. It nested and foraged in groups, and some researchers have argued that the social structure of the species may have made it more vulnerable than its raw numbers suggest. Once colonies fell below a certain size, the argument goes, the birds may have lost the ability to find mates and locate food sources efficiently. The genomic evidence does not resolve this. The mechanism is still debated. What remains true about Martha is plain. She was a female passenger pigeon. She spent most of her life at the Cincinnati Zoo, where she had been since at least 1902. She was old for her species, somewhere around 29 years when she died, though the records on her hatching date are not precise. A zookeeper found her on the floor of her enclosure on the afternoon of September 1st, 1914. The cause of death was not recorded with any clinical detail. Old age is the usual assumption. She was not a genetic anomaly. She was not the weakened survivor of a population already in collapse. The genome extracted from her remains, and from the birds in the museum drawers beside her, look like what you find in a species that was large, stable, and healthy, and then was not. What exactly made 3 to 5 billion animals disappear in 50 years, the full mechanism of it, the ecological cascade, what the loss of 40% of North America's bird life did to the forests that shaped their existence for centuries, is still not entirely resolved. Bob Zink is still saying so. The question remains open. Martha is at the Smithsonian. She has been there for more than 100 years. She is not hard to find.